All right, hello everybody. My name is Katsarakes, and today I'm going to be talking to you guys about my senior design project, which is boosting the chemotherapeutic effect of cancer drugs using focused ultrasound. And this was advised by Dr. Yu at the Neuromodulation and Tissue Engineering Laboratory. So first, I'm going to give you guys a bit of background on why this project is significant. So as you all know, cancer is a huge problem in the United States and around the world. And in fact, 39.5% of all individuals in the United States will be diagnosed with some form of cancer over the course of their lives. So naturally, the scientific community has invested a great deal of resources into the prevention and treatment of cancer, and this has resulted in hundreds of drugs on the market, each with their own treatment methodologies and targeting different types of cancer. A lot of these are systemic therapies, which means they travel through your bloodstream to get to the site of the tumor. And one issue that these systemic therapies often have in common is that they have interactions with blood plasma proteins. And these interactions mean that the blood plasma proteins hold on to the drug molecules and sequester them in the bloodstream, and they're not able to travel to the site of the tumor. So as you can see in this figure, the drug molecule we've chosen to analyze is cisplatin, which is a treatment for cervical cancer, and it interacts with a protein called albumin. And the albumin molecules bond to this drug molecules in the bloodstream and prevent them from traveling to the tumor, which is obviously a big problem and can reduce the efficacy by as high as 95%. So we definitely need to come up with some strategies to mitigate this issue. And something you might consider is just giving a bigger dose of the drug, because that means that more molecules will travel to the site of the tumor. However, this is a problem for chemotherapy because these drugs are associated with severe side effects, and giving a bigger dose is going to mean more severe side effects, which can be life-threatening. So you might also consider some other methods, such as advanced drug delivery techniques. For example, you can wrap the drug molecules in a nanoparticle and try to get them to the site of the tumor without any interactions with the blood plasma proteins. But in general, these methods are typically expensive and difficult to manufacture, and it can take years to research. So we were interested in a technique that was cheaper and was much more easy to implement. And so we came across our tool called focused ultrasound. And the principle of this method is using low-intensity acoustic pressure waves to essentially mechanically vibrate the blood plasma proteins at the site of the tumor and help them to shake off the drug molecules in order to allow those molecules to travel to the tumor. And so you can see the three main points of improvement for this method over chemotherapy alone is that when you apply focused ultrasound, it's low intensity, it's non-invasive, and it's spatially selective. So what's important about these things is, firstly, low intensity means that it's not going to cause any damage to nearby tissue. High intensity waves can sometimes elevate the temperature and cause burns, or it can just be damaging to nearby healthy cells. Our method is very low intensity, so it's not going to cause any of those issues. It's non-invasive, which is really important because a lot of surgical techniques require invasive surgery, which can be very traumatizing for patients. And this is very easy to implement because we don't have to do any surgery. You just apply it to the site of the tumor. And finally, spatial selectivity means that we're only increasing the drug concentration at the site of the tumor rather than increasing it across the entire body, which would obviously result in higher side effects. So the whole point of this method is to achieve increased drug efficacy by unbinding the drug from the blood plasma proteins at the site of the tumor, and to reduce the necessary systemic dose of the drug that's needed to provide the same level of treatment. So an overview of how this project worked: first, we had to actually come up with the method to apply focused ultrasound. Second, we needed to use an in vitro experiment, which basically means doing testing in the lab, to figure out whether this method was functional. And finally, we used an in vivo experiment, which means testing in animals, to figure out if this method was actually going to work biologically. So the first thing that we did was to actually generate our technique. And obviously, this came with a lot of details about how to actually produce some focused ultrasound and apply it to a site on the body. So there were a lot of details that I'll spare you, but they can really be sorted into two different categories, which is calibration, which is making sure all the components work as they're supposed to, and then characterization, which means testing our system with a bunch of different inputs, for example, different amplitudes of our ultrasound frequency, and making sure we get the expected output at the end. And so once we had done this, we were able to move on to our second aim for the experiment, which was doing the in vitro testing. And for this, we used a method called equilibrium dialysis. And essentially what that means is we had 3 cassettes filled with fluid, and this fluid contained blood plasma proteins and cisplatin molecules. And with one cassette, we placed it directly in line of ultrasound waves. With one cassette, we placed it slightly out of the line of the ultrasound waves. And with the third cassette, we placed it very far away as the control group. And we saw from this method a very successful outcome. And in fact, we saw over 25% reduction in the cisplatin unbinding compared to both of the out of focus and the control group, which means that it's only affecting locally and it's not causing any changes outside of the focus of the sonication. And so once we were able to confirm these results, we were able to move on to our in vivo experiment. So I produced for you a diagram of how this experiment went. Basically, in essence, it was injecting mice subcutaneously, which means under the skin, with human cervical cancer cells, because as you know, cisplatin is a treatment for cervical cancer. So we needed to use these mice as a model for the correct type of cancer, which was human cervical cancer. And once these cells had been injected, we gave them 4 weeks to grow to an adequate size. And then we intervened with either cisplatin treatment by itself, cisplatin and focused ultrasound treatment combined, or no treatment at all. And we did this for 2 weeks, intervening 2 times a week. And finally, we analyzed the reduction in tumor volume at the end of that period. So I've produced for you here a visual of the experimental design. So as you can see, we anesthetized the mice using isoflurane gas, and once they had been put to sleep, we were able to give them an intraperitoneal injection, which means into the gut cavity, of cisplatin molecules. And once that had been done, we were able to apply our focused ultrasound using a waveform generator, which produces the signal, porting that waveform into a focused ultrasound transducer, which produces the mechanical vibrations, and then sending those vibrations through a soft gel called a coupling hydrogel to apply it directly to the skin at the site of the tumor. And so you can see some real-life pictures. At the top is our waveform generator, which created the signal, and on the bottom you can see the mechanical arm we designed to apply the focused ultrasound, porting through the hydrogel to the surface of the skin at the site of the tumor. And we applied this treatment for 60 minutes, as I said before, 2 times a week for 2 weeks, in combination with the cisplatin treatment. And finally, we were able to observe the results at the end to determine whether or not our method was successful. So in the end, we saw some extremely promising results. As you can see on our graph, the cis plus and FUS positive group, which means the mice who received both cisplatin and focused ultrasound, had a much, much better response to the tumor compared to the mice who only received cisplatin and compared to the mice who received no treatment at all. So you can see on the graph, the mice who received no treatment, their tumor continued to grow throughout the duration of the treatment period. For the mice who received only cisplatin, their tumor growth plateaued. And for the mice who received both cisplatin and focused ultrasound, their tumor volume decreased throughout the duration of the treatment period. So some takeaways I want to draw your attention to is firstly, that the precise results we determined was that we had a 68.9% decrease in tumor volume for the cis plus and FUS positive group, which was much greater than any results that had been seen in the literature previously using this technique with other drugs. So we were very excited about that result, especially relative to the 7.2% increase in tumor volume for mice who only received the cisplatin treatment. So as you might expect, it was surprising for us to see that even giving a well-known, very useful chemotherapy technique didn't even decrease the volume of the tumor. It still resulted in a slight increase compared to our method, which significantly decreased it. So the next things we have to do are some technical details. First, we have to make sure that there's actually a lot of cisplatin inside the tumor. We have to quantify how much cisplatin has actually made it in there. We have to make sure that there's no increased cisplatin concentration throughout the plasma of the body, again, to ensure that this is a local technique and it's not elevating cisplatin levels throughout the mouse's body. And finally, we need to expand the study to additional chemotherapeutic drugs to make sure that this interaction works with a variety of drugs and different blood plasma protein molecules. So future directions for this research is that we are eventually trying to clinically translate this technique to be used in conjunction with chemotherapy on human patients and hopefully help them reduce the systemic dose and reduce this necessary suffering for them to achieve the same degree of treatment by giving them a lower dose of chemotherapy and preventing them from experiencing severe side effects. So we're very excited to publish this work in the near future. I'm thankful for you guys for listening to this. Thank you.
Perry, that's like a crazy reduction in the tumor size. Just curious, in terms of, were there concerns about side effects or going into it saying, hey, these are the things that we also want to look for or watch for, or just like where was that on the radar screen?
In terms of side effects, so when you're doing studies with mice, obviously it's hard to determine exactly what kind of side effects they're experiencing. So we analyze the body weight and also the behavior of the mice to ensure that they're not experiencing any significant changes. And naturally, with any dose of cisplatin, because the mice are so small, they're going to experience some side effects. But we did a control group naturally for testing with only focused ultrasound and no cisplatin. There was absolutely no change in the side effects that were experienced by the mice undergoing focused ultrasound. So that was a very positive outcome.
Great talk, Perry. I was curious about the coupling hydrogel. If you could elaborate a little bit more on how that targets, I guess, the ultrasound to the right spot, or what its purpose is?
Yeah, so like I said, the transducer is what produces the mechanical vibrations, and those vibrations are sent through a PVA gel, so we manufactured that, and it's just a soft gel in order to make contact with the skin, because you can't do that with a hard material or it would cause abrasion to the skin. So we use a soft gel so it can make direct contact with the skin, and then we put some like ultrasound gel at the surface of the skin to lubricate it, and that's just what sends the mechanical vibrations through to the skin. And as you can see in the figure, it sends the ultrasound waves to the side of the tumor so it can unbind the drug molecules right there.
Really, really interesting work. I have a question about, I'm glad you're on this slide, the in vivo segment of this, which has to do with, from my sense of it, you're using the mouse basically to inject this very particular kind of human cell, right?
Mm-hmm.
And then doing the experiment and the therapy from there. I'm wondering what the introduction of the tumor at this point, of being able to grow what would be normally in a human vessel, the tumor there, has in terms of the overall mouse physiology, and whether there are other factors that you take into consideration, how much you can really isolate this once it's part of a mouse's body, and then what that does in terms of your thinking about the effects of this. I mean, the effects are amazing, but then how do you think of that in terms of the mixture here?
That was part of the reason we chose a subcutaneous injection of the tumor cells, because it literally grows the tumor at the skin, so it's growing upward from the skin. It's not growing inside the mouse's body, and that was twofold. It allows us to measure the tumor very easily, and it also allows us to apply the sonication very easily. And naturally, there are some things you might expect. For example, ultrasound is known to increase blood flow, so there are some additional experiments we might need to do. An example is we have a technique called laser Doppler flowmetry that can analyze blood flow increase, and we can kind of tease apart those effects to make sure that it's the focused ultrasound that's actually causing the increase, and not just like an increase of blood flow drawing more cisplatin molecules. So those are things that will need to be done, but it still is very promising results, and we're excited to continue this research in the near future. Thank you.